Hello to everyone on their screens, and special thanks to Vito for inviting me to this exciting webinar. My name is Labscale. I'm a biologist, managing director, co-inventor and co-founder of CellDEG. Since 2011, my colleagues and I have been developing membrane-based cultivation systems to enhance productivity and finer cell density of photoautotrophically grown cyanobacteria and microalgae. We wanted to create flexible systems with interchangeable platforms, one that can provide homogeneous growth conditions for comparative studies, but can also be used for larger volumes to produce biomass efficiently. In 2013, we had a major breakthrough by discovering features that enabled highly productive growth to extremely high cell densities. These findings led to the founding of CellDEG in 2016 as a spin-off from the Humboldt University of Berlin. It's our vision to establish CellDEG cultivation systems as a new lab standard to make results comparable across lab boundaries. Furthermore, we believe if we apply this technology in 10 to 20 liter modules, we can easily outperform 100 to 200 liter PBRs in terms of productivity. What distinguishes CellDEG PBRs from existing solutions? The principle of a conventional bubble column photobioreactor uses CO2-enriched pressurized gas for mixing and as a feed of carbon dioxide to the cell suspension. It is a well-known method which works for growing microalgae strains but has certain disadvantages, like most of the injected CO2 is wasted to the atmosphere. Strong bubble streams induce shear stress to the cells, and light penetration is limited due to larger diameter of the tube, which limits volumetric productivities and finer cell densities. And when used for comparative studies, it poses a challenge to achieve equal mixing by adjusting bubble rates. In contrast, the CellDEG cultivation systems use nanoporous hydrophobic membranes for bubble-free CO2 supply. The cultures are turbulently moved by an orbital shaker, and here the light path length is below 10 mm and could be easily adjusted by the applied volume. Because of the consistent turbulence, CO2 supply and irradiation, this technique allows for highly productive screening platforms with many parallel replicates. In the following slides, I will explain how this technique works and why it is beneficial for many users. This slide shows a schematic design of a CellDEG cultivation system based on the high-density cultivation technique. Let me start with a CO2 compartment of the cultivation platform, which is connected to the growth control unit, or GCU, via a forward and a backward flow, forming a closed circular gas loop which is separated from the atmosphere. The air-CO2 mixture ratio is controlled by the GCU, which increases the CO2 concentration in the gas loop by injecting small amounts of pure CO2, which can only escape by diffusing through the basal membrane into the thin layer culture, causing a negligible loss of CO2 during cultivation. This also allows online monitoring the biomass growth by counting the introduced CO2 volume. At the top of the cultivator is a vent membrane serving to exchange produced oxygen for atmospheric nitrogen. Above the cultivation platform there is a high-power grow LED with intense light emission and low heat dissipation. The software of our growth control unit allows for programming light intensity and CO2 partial pressure during cultivation. In addition, it has important recording and security functions. On the next slides, I would like to showcase our available products. There are two control units, the very flexible GCU1, focusing more on laboratory applications, and the robust GCU Pro, which we developed for larger scale biomass production. All shown cultivation platforms can be connected to both control units. We offer cultivation platforms for up to 45 times 10 ml cultures, up to 9 times 100 ml cultures, or one single unit with a capacity of 1.5 liter in which biomass growth can be monitored online via the amount of CO2 absorbed by the culture. This is our high-range transmission turbidity sensor, which we developed for precise and reproducible readings of the optical density in an extremely wide measuring range, or for example, if calibrated to OD at 750 nanometer, from 0.1 to more than 50. The sensor is used non-invasively and measures directly in the HD10 and in perspective also in the HD100 cultivators, so no sampling or dilutions are required. From here I want to focus on the main areas of application in which the CellDEG HD systems offer unique advantages. First, the screening platforms in 10 and 100 ml scale allow a large number of replicates to grow rapidly and under homogeneous conditions. Using the HRTT sensor, we are able to compare individual growths and create growth plots very efficiently, even in 10 ml scale. Examples for application would be optimizing productivity by comparing different growth conditions or media, screening novel strains for natural compounds, or the selection of mutants. Second, various studies already demonstrated that with the CellDEG high-density cultivation technique, not only more biomass could be produced, but also that secondary metabolites, storage polymers, or terpenes produced by genetically engineered microalgae were present in much higher concentrations, making it an outstanding production system for high-value compounds in lab scale. I will provide some examples in the following slides. Third, our production platforms offer a significantly faster cultivation of photoautotrophically grown exenic and vital biomass. This biomass can be used to inoculate larger photobioreactor systems for scaling up your production. This figure shows the increase in optical density of Cyanocystis and Nanochloropsis salina in CellDEG HD media in 10 ml scale at 32 degrees Celsius with 4 replicates each. Additionally, the programmed course of the photon flux density and the CO2 concentration is plotted. We observe rapid growth with a long linear light-limited phase and low standard deviations for both organisms. In case of Cyanocystis, we obtained an OD of nearly 90, corresponding to around 28 gram dry weight per liter within less than 6 days, starting with an OD of 0.2. In a paper of a work group in Potsdam, Germany, published in 2017, the biomass yield of nitrogen-fixing Nostoc punctiforme, cultured in a conventional system and in an HD system, was compared. After 24 days, even without optimal conditions, around 400 gram wet biomass could be harvested from high-density systems, compared to only 15 gram from the conventional cultivation, demonstrating a 26-fold increase of biomass yield in an HD system. In this paper, the same group cultivated Nostoc punctiforme under nitrogen-fixing conditions again to compare natural compounds in the biomass of an HD cultivation to the conventional cultivation via HPLC. The blue line represents the HD culture, the red line the conventional culture. The difference in the numbers and amounts of compounds is quite remarkable and speaks for itself. Results showed an upregulation of more than 50% of biosynthetic gene clusters for the high-density cultivation. A group in Uppsala, Sweden worked with one of our starter kit systems with a bicarbonate buffer as CO2 source and compared it with a reactor comprising several parallel bubble columns by growing genetically engineered Cyanocystis to produce terpenes. They found that in the same time the bubble column reached an OD of nearly 3, in the HD system an OD of around 45 was obtained. The bisabolene titer was more than 100 times higher than previously reported. Even the titers of patchoulol and bisabolol were many times higher than in the reference system. This figure simply demonstrates the increase in OD over time of a Cyanocystis and an Arthrospira maxima culture at 32 degrees in one volume of 1000 milliliter and the programmed course of the PFD as well as the CO2 concentration. Both organisms could maintain a productivity of more than 5 gram dry weight per liter a day for several days in batch mode. After only 7 days of exenic cultivation, a finest cell density of around 21 gram dry weight for Cyanocystis and 28 gram dry weight for Arthrospira was obtained, which could be used to inoculate bigger reactors. Let me summarize why the CellDEG systems are perfectly suited for investigating the full potential of photoautotrophically grown microorganisms. We are able to cultivate exynically with productivities of up to 10 gram dry weight per liter and day, to cell densities of more than 30 gram dry weight per liter in batch regime using CellDEG HD media. The systems offer homogeneous cultivation conditions enabling great reproducibility. Our non-invasive wide-range turbidity sensor reduces time for OD determinations and allows to prepare growth curves in 10 ml scale. Online biomass monitoring by counting consumed CO2 volume is possible by cultivating larger single volumes. The cultivators have low water vapor and negligible CO2 losses. We observe very high quantum yields of around 1/3 to half the theoretically achievable maximum, even in strong light at high cell densities. The systems offer great flexibility through interchangeable cultivation platforms and have low safety requirements, as they don't need big and potentially harmful pressure cylinders, because it can be operated with a 0.5 kilogram gas cylinder for around 4 to 5 months. In conclusion, the CellDEG system offers a number of key benefits, including highest productivities, ease of use, reproducibility, safety, and the ability to cultivate to extreme cell densities under optimal conditions.